All right, guys, picking up from the video that we saw in class, let's take some notes on a trophic cascade. So you saw in the video that we watched today this sort of ecosystem that involved initially starfish and their the things that starfish ate and how those starfish impacted the number of organisms that could live in a system. And then we saw a second kind of study, an extension, that looked at kelp and sea urchins and then also sea otters, right? And so these two pictures will help guide our conversation here on this page. So a trophic cascade is something that happens when there is a very large disruption to the energy distribution in an ecosystem. Usually it involves a loss of a keystone species. Now, a keystone species we saw in our video is really named from an architecture concept, which is as you build an arch, you build up two sides of an arch and then you lean those two sides together onto a keystone. And so this keystone really holds the arch together. And if you remove that keystone, the arch will collapse. Well, a keystone species is a species that's in a system that really impacts that system greatly, to the point that if it's removed, the whole system could collapse. And so we say that a keystone species has a disproportionately large effect on the overall system. If you remove these species, oftentimes you can see a collapse of an entire system. And if not a collapse, then a pretty significant impact or change in the system. So a lot of times the organisms that are removed when a trophic cascade occurs, the organisms that are removed are keystone species. And if that keystone species, as they often are, is an apex predator, it causes something that the video talked about. It causes a top-down trophic cascade, which means basically removal of or increase of this top apex predator, which is typically a keystone species, greatly impacts the levels below. The interesting thing is it impacts each level. It sort of toggles back and forth between, if, let's say, for example, when the sea otters were gone. So in the study that they were looking at, they went into a tide pool where sea otters were not present. And the loss of the sea otters made the sea urchin population grow unchecked because the otters weren't there to eat the urchins. Well, a decrease here led to an increase here. And with all of those sea urchins present, they ate all the kelp. And so we see a decrease in the kelp population. So, hold on.
Teachers and students, if you are parked in the blue zone, the marching band zone outside, please move your cars to a visitor stop. Thank you.
If you ever wonder the announcements in the building when you all leave for the day, it's stuff like that. So you have a decline here leads to an increase here, which leads to a decline here. So negative, positive, and it would just kind of keep toggling down. So each level is inversely affected than the one above it in a top-down cascade. Now we could flip the script here and we could talk about a population explosion of sea otters. So there's so many sea otters that they eat up so many of the urchins, and then the urchin population declines. What does that do to the kelp population? It shoots up because there's no natural predator then to eat the kelp. So trophic cascade is sort of an interesting concept that talks about controlling. Prior to this understanding, we thought the primary way to control a food chain was at the base level, control the number of producers, and that controls everything. Well, now we can see that in addition to controlling producers, we can also control from the top down. All right, we are taking a turn into the world of photosynthesis, right? So let me blend this together. We've been talking energy in an ecosystem. We know that energy is created at the base level of a trophic pyramid by producers. Producers are going to take a form of energy that's not really usable to us in biology, in a life food chain system and convert it into energy that is. And so the primary chemical reaction that we're going to look at here is what's going on in the process of photosynthesis. So photosynthesis is our focus. You do need to have a little bit of understanding about a leaf, plants in general, the structure of a leaf, and then these things called chloroplast. So let's start with the leaf is the primary site of photosynthesis in a tree. And some of you might go, well, yes, that's where all the green parts are. That's true. Leaves do have chlorophyll in them, and they are big and broad, and so they have lots of surface area, most of them, and they are at the tippy top of the plant, capturing as much sunlight, open, nice and big, capturing as much sunlight as possible. So other interesting things, because it's the site of photosynthesis, because there's so many chloroplasts there, carbon dioxide must be able to enter the leaf. And then through the process of photosynthesis, we're going to make a little oxygen, which actually is like a waste product. It's not the goal of photosynthesis, but it's a waste product. That oxygen has to be able to leave the cell or the leaf and enter the atmosphere. Water is also the final kind of stopping point. The leaf is the final stopping point of water. So we know that water moves into a tree or a bush through the roots, and then it travels all the way up to the leaves. That water is going to be used in the process of photosynthesis. So this is kind of the ultimate destination for carbon dioxide, oxygen, and water. We get all those things together inside of a leaf. If we have light and the chloroplast, we can make some sugar. As a reminder and as a kind of throwback and review to our water unit, movement of water up to a leaf, like movement up the tree to a leaf, that is called transpiration. It happens because of the joint action of adhesion and cohesion. All right, so let's talk structure of a leaf. There is a diagram in your notes that looks a bit like this. And so we're going to label that diagram. And as you label the parts, you're going to write a little something about the parts under it. So we'll start with the two large layers that exist within the leaf. So the big chunk of the leaf here in the middle is broken down into two layers. Each one is called a mesophyll. So you have the top layer up here with all of these tall cells called the palisade mesophyll. So bracket this off and label this as the palisade mesophyll. And here are a few things about the palisade mesophyll to write underneath your labeling. This is the top of the two layers, and you can see the cells are really long and they're very tightly packed together. They have tons and tons and tons of chlorophyll in these cells, which is perfect because think about, they're at the top of the leaf. Where's the sun going to hit the leaf? On the top. And so here are these chloroplasts that are ready to receive that light and start the process of photosynthesis right away. The other layer of mesophyll is this sort of bubbly layer that you see down here. So bracket this out and call this the spongy mesophyll. The spongy mesophyll has really loose packed cells, and that's on purpose. A second ago, we said that carbon dioxide needs to enter the leaf and oxygen needs to leave. Well, that enter and exit is happening through these tiny holes that we'll talk about in a second on the bottom. And so the gases come in and out. They have to kind of work their way through this area. And so if the cells are loose, not densely packed together, those gases can move around more freely. There's also something called a vascular bundle in this spongy mesophyll. We're going to come back to the vascular bundle and talk about that. And so you can label it in just a second. Moving forward, the next big things we need to label are our epidermis. So you have two epidermis layers. You have an upper epidermis, which is right here on the top, this slightly less green or dulled green layer. And then you have a lower epidermis. So if we talk about the upper epidermis for a second, notice that on top of the epidermis is another layer called the cuticle. Label the cuticle. The cuticle is a big waxy layer right on the surface of the top part of the leaf. And that cuticle is actually going to help the plant to not lose water back out to the atmosphere. So we know that water comes up the roots and up the stem or the trunk of the tree into the leaves. Well, that water needs to hang out in the leaf long enough that photosynthesis can occur. And so we don't want that water to evaporate straight back out into the environment right away. We want to keep it in the leaf. And so this cuticle helps to do that. It helps to prevent water loss in plants. The lower epidermis doesn't have a cuticle, but it does have these little tiny holes. And on your diagram, they look kind of like macaroni noodles. Blown up, they look like this. You're just seeing one of these cut in half because it's a cross section of a leaf. Those are called stomata, and we're going to talk about stomata in just a second. The next big structure to label is the vascular bundle. So over here in your diagram that you see the word vein, under it vascular bundle, this whole thing is the vascular bundle. It's made of two systems of tubes. So one tube system is called xylem, and the xylem tubes inside the bundle are going to move water. And water really only moves in one direction in the plant. It moves from the roots of the plant all the way up to the leaves. So the xylem tubes are like a one-way street. The phloem tubes are the second type of tube system here in this bundle, and those are going to move the food or the sugars that are created in the process of photosynthesis. Those go everywhere. So once the plant creates sugar, it's going to use some of that sugar to support its own metabolic needs. But then many plants will store sugar in fruiting bodies. So when you eat an apple or when you eat a carrot, that is the fruiting body of the plant, and it's loaded with the sugars that were produced as a part of photosynthesis. So these phloem tubes are directing those sugars to storage sites, either the actual tissue of the plant to make the plant get bigger, or to the fruiting bodies of the plant that we get to come along and eat. So make sure the vascular bundle is labeled here in your diagram as well. The last thing I want to talk about is a stomata. Stoma is like one, just talking about one of those. If you're thinking, well, why is this? That is a different word. Stomata are like little tiny pores in the lower epidermis. And these pores allow carbon dioxide to come in and oxygen to come out. The pores also can let water vapors escape, though. So I tell you that because it's a trade-off. When the pores are open, when the stomata are open, the plant is pulling in tons and tons and tons of carbon dioxide for the process of photosynthesis. But it's also losing water vapors, which is a little bit dehydrating to that plant. So some plants, especially those that live in really warm environments, desert environments, dry environments, they have strategies to open their stomata at certain points in the day to minimize the water that is lost. Now, these holes, the pores, are flanked by cells, and those cells are called guard cells. And so if we look at this picture down here, this is a guard cell, and this is another guard cell. The hole in the middle is the stoma or the stomata. The guard cells are completely controlled by how much water they take in and store in these vacuoles. So it's a great connection back to osmosis. When those cells swell up with water, when their vacuoles are full with water, the cells sort of pucker open and create the stomata hole. So full guard cells equal open stomata. When those guard cells don't have a ton of water in them, when they're all shriveled up, they collapse back and they don't allow a hole to exist. So full guard cells full of water equals open stomata. Empty guard cells, no water, equals a closed stomata. So those are completely being controlled by water coming in or coming out of the cells. Next thing I want to look at is a chloroplast, and we've talked about chloroplast before, so I don't know that a ton of this is going to be so new to you. These are our double membrane bound organelles. I toss out a thanks to the endosymbiotic theory. If you don't know what that is, you should go look it up as we are preparing for this AP test. That's something that sort of gives us an idea of how complex cells evolved. So check that out. But long story short, chloroplasts have two membranes. There's an inner and an outer membrane. So on your diagram, pause for a second here and label the inner membrane, which is kind of light green in my picture, and the outer membrane, which is dark green. We also need to know that the inside of the entire chloroplast is filled up with a fluid, and that fluid is called stroma. So make sure that you label the stroma inside. And then we need to talk about these stacks, these towers. So a grana is one stack of a bunch of thylakoid discs. So make sure that you've labeled the grana and the thylakoid disc on your diagram before moving forward. Now, the discs are so important because the discs have a membrane, and in those membranes are the pigments, chlorophyll A in particular, that is able to capture light energy and start the conversion process in turning that light energy into chemical energy that can enter a food chain or an ecosystem. So without chlorophyll, without something that's capturing the light energy, we can't start that conversion process. So thank you to the thylakoid discs that have chlorophyll A embedded in their membranes inside these individual chloroplasts. Now, I tossed out chlorophyll A. A is one type of chlorophyll. It is a chlorophyll that makes plants look kind of green, which really means that it's good at reflecting green back to our eyes. And so we see chlorophyll as a pigment as a green color. I'm going to talk more about that in just a second. There are other types of chlorophylls and plant pigments as well. There's also a B, which is chlorophyll B. It's also green. It's a little bluer green in coloring. And then there are some complex words that really I chose to just collectively call accessory pigments that are orange and yellow and red that exist within these chloroplasts. So that's really what's responsible when fall hits and the leaves start to change colors. They really aren't changing colors. Those colors are in the leaves all the time, but the chlorophyll A and B is so pronounced that it's covering up the orange and the yellow and the red. So when the temperatures change and the chlorophyll A and the B start to break down and that green goes away, we get to see those other pigments that are in the leaf for a short period of time, and then they break down as well, and the leaf goes into, or the tree goes into hibernation, the leaves fall off the tree. Last thing that I want to talk about, which comes back to what I dropped just a second ago, that chlorophyll A is really good at reflecting green light back to our eyes. We need to talk just for a second about the electromagnetic spectrum and light. So this is the picture that's in your notes. This is a picture of something called the EM, or the electromagnetic spectrum. The electromagnetic spectrum is an energy spectrum that shows you the length of wavelengths and frequencies of wavelengths of different types of energy. And so really what we're looking at here first is that short waves have more energy in them. They're tightly packed together. And so things like gamma rays and X-rays are loaded full of energy. Really long waves have less energy in them. What's important to us for photosynthesis is what sits right here in the middle. This is the section of the electromagnetic spectrum that refers to visible light waves. So what we can actually see. We cannot see radio waves. We cannot see gamma rays, but when they hover around this wavelength, we can see them. And so visible light, which is what is collected and used in the process of photosynthesis, is on the spectrum. It's a very small range of the overall spectrum. I mean, look at that. All of that right there in comparison to the entire spectrum, it's a small section of the spectrum. And if you blow that up, you know that visible light, which looks white to us, actually is made up of a collection of colors. So think about a rainbow, right? You probably learned the acronym ROIGBIV: red, orange, yellow, green, blue, violet, or green, indigo, violet as we progress. So the red spectrum is a little bit larger wavelengths, and the blue down here is a little bit smaller. So a little less energy in red and a little more energy down here in blue. Each color, what's important about that, each color of visible light corresponds to a different wavelength value. And so when we are going to talk about photosynthesis, we'll talk about when light hits an object, like a leaf, and really any object, there are a couple of things that could happen to that visible light. One, it can go straight through the object. That's called transmission. And here is transmission. The light goes through, it is transmitted through the object. It could be absorbed by the object, which is what we see here in the middle. If it is absorbed, no light goes through, no light bounces off, it is truly absorbed. Absorbed light is the light that is captured in the process of photosynthesis. The third thing that could happen is light could hit the object and it could bounce off of the object. So that's when light is reflected. And so for us, here's where it matters. Light that is absorbed is used in photosynthesis. Colors that are seen to our eyes indicate that when light hit that object, that particular wavelength of light bounces back to our eye, and that's how we perceive that color. So here's an example of that. If I shine visible light on a yellow surface, the surface is either absorbing or transmitting red, orange, green, blue, indigo, and violet, but it is reflecting yellow back to our eyes, which is why I see this surface as yellow. So when we look at green plants, things that are green, it's green because that's what's reflected back to our eye. So wavelength hits a leaf, and visible light hits a leaf, and the portion of visible light that's around 500 comes back to my eye, and I see it then as a green leaf. So we see the reflected light, that's like the color that we see. So whatever wavelength is reflected back to us corresponds to a particular color. So if you are holding a pen or a pencil in your hand, like I'm holding a pen right now and the pen is blue, that means that when visible white light is hitting this pen, all of the not blue values are either being absorbed by the pen or passing through the pen. But what exists at this particular blue value is reflecting back to my eye, and that's why the pen looks blue to me. You might be going, what in the world is happening, Miss Mac? Well, we're building towards an understanding of this graph right here. This is an absorption spectrum, and this happens to be the absorption spectrum of chlorophyll A. So let's talk about an absorption spectrum. Absorption spectrums show the amount of light that is absorbed by an object at various wavelength values. So here's what I mean by that. This, which I already told you is chlorophyll A, is really good at absorbing light waves between 4 and 500 nms. It's also really good at absorbing light waves between 6 and 700. It is not good at absorbing light around this value. And so this is what is reflected back to us and is why chlorophyll A looks green to our eyes. Okay. The important thing to know is that the more wavelengths you can absorb, the more photosynthesis is going to really occur. So chlorophyll A actually can absorb a whole bunch of wavelengths related to all of the visible light spectrum. It's just not great at absorbing these. And so these are reflected back to us. But it's really great at absorbing here and here, which is probably why chlorophyll A is the primary pigment in photosynthesis, because it's so good at capturing lots of different wavelengths of light for that process of photosynthesis. Specifically, I want you to know about this spectrum because it has cropped up on past AP tests. The chlorophyll A absorption spectrum shows us that A is good at absorbing all colors pretty well, except those that exist in the blue-green color wavelength, which is what is reflected back to us. And that's why so many plants look green, right? So that means that we see chlorophyll as blue-green, and I'm not sure what I dropped off there. We see chlorophyll as blue-green, and it totally went away. What was I going to say? Oh, and it is the primary pigment in photosynthesis because it's so good at absorbing all of those other color range values. So here's an example, and this is where we're going to end. This was a 2013 FRQ, not the questions over here, but this data and graph was given to you. So we're actually going to look at that FRQ in class. So I don't want to spoil the whole FRQ, but I do want you to see what we're looking at. So here's graph 1, which is an absorption spectrum. And it looks like in graph 1, we see a peak right around here of absorption. So you have to know, what does absorption mean? What does absorption tell us about the organism or the plant that, you know, is using the light that's being absorbed? Here is graph number 2. And then down here, it gives you the actual wavelength values associated with the colors. So violet is from 380 to 450. So the violet blue range would be here, and we would progress down here to the red. So my question to you is, in graph number 1, what color do you think that this organism would look like to us? So think about it for a second. What if this organism showed this particular absorption spectrum, what color do you think that it would look to our eyes? All right, so think about a little bit of a guess here, hopefully not a guess, but an actual understanding. This organism is going to show us the colors that it's not good at absorbing, right? Because if it's not being absorbed, it's probably being reflected back. So this is really good at absorbing values between 500 and 650, which is anywhere around this kind of transition from green to yellow to orange to red. So this organism is probably some kind of bluish purple organism. This one is what color? That one's green, right? We just saw this one. This was chlorophyll A's absorption spectrum. So it's really good at absorbing this. It's really good at absorbing this. Is not so good at absorbing things here around 500. And so what's hanging out around in the 500s? That's bluish green color. So this one's probably green, blue, green, green. If you wanted the maximum amount, I'm going back over here to question number 2. If you wanted the maximum amount of photosynthesis possible, which organism would you choose? So think about that for a second. Remember, high absorption indicates higher rates of photosynthesis. If I chose an organism that is going to produce the most, like photosynthesize the most, I'm going with the organism that's represented over here in graph 2, because they can absorb so many different color values here and here, indicating they're really good at capturing multiple different wavelengths of light. So we'll look at that question, the actual question, but that's how you would read that particular graph related to photosynthesis, related to light and energy and the ability to absorb that energy. Okay, when you're done, you are done.